Hi, everyone. Welcome to this session on digital data and new age of fisheries analysis. We decided to give this brave title. And we heard a lot of talks about the challenges for small-scale fisheries, some opportunities also, but mostly a lot of challenges. And one of the challenges that we heard quite often is that there is no fish. And so this session takes a lot of mostly biological perspective, not so much socioeconomic, but biological perspective, so that if there is a lot of fish, then fishers are also going to do well in most cases. But we want to know, is there lots of fish? Is it getting better? Is it getting worse? And in most cases, we hear that small-scale fisheries are very data poor. So like we have heard that nearly all small-scale fisheries are data poor. We don't know, is there lots of fish? Is there not? However, if you just Google small-scale fisheries, that's the first thing that came up. The first thing you see is lots of people. And if there are so many people, it's people everywhere. And if there are so many people, then there is a lot of knowledge. You can't have data poor fisheries when there are so many people watching all the fisheries and all the fish. So perspective of this session is that there's actually a lot of data, there is a lot of knowledge. We just need to access it, share it, and use it in the best way. And of course, that's where we need digital tools, modern technology, and people engagement. But what kind of data should we collect? What sort of data is needed? There are a lot of questions. How to collect it best? Who owns the data? How to share it? And we will hear a lot of interesting talks about it in this session. I can't help myself to talk about fish size. This is the topic of my research for a long time. And I would say that data on fish sizes is extremely important. It's important for humans. It's important for fish. It determines population resilience. It determines reproductive capacity. It determines the role in the ecosystem. And people like big fish, it affects their market values and all sorts of other things. And it's actually nice to look at beautiful fish, even if you want to just watch them. The good thing why we need size data is that even small amounts of size data can actually tell us about something about population status. So let's say we have these two samples from two different populations. One is orange, the other one is gray. And using some statistical methods, we can make some predictions about how the size distributions in these populations look like. And we can already say, okay, the population that has more big fish probably is doing better than the one that doesn't have all these big fish. So it's easy to collect the size data. It's easy to understand. It's easy to communicate. We should do it. Another reason why we should do it is that fish sizes are also changing very rapidly. There are two main reasons for this. One is fishing. Of course, we just removed all the big fish. There is none left. But increasingly, it's also climate change because temperature really has a big effect on fish sizes. But unless we collect this data, unless we watch it, we won't know. One interesting example is these historical fishing competitions, for example. And so there is a study that showed the trophy fishing competitions in Florida, where in 1958, the winners had the fish that were almost bigger than people. In 1980s, they're about half the size of people. And in 2007, they looked tiny. But the interesting thing that's in every case, people thought they caught really big fish and they were really happy. But because they kind of forgot how it actually looked, they don't maybe even know that information, how it could have been even just 60 years ago, 50 years ago. So I would say that not collecting fish size data is a hugely missed opportunity. For example, there is a platform, iNaturalist, which involves a lot of people. And if you just go there, we have 1 million observations of fishes from all around the world. But there is almost no size data associated with it. So that's a hugely missed opportunity. You can't use that data really for population analysis. So many questions about what sort of data, who owns the results. That's very important. Who shares them? Who manages the data? And how to use this data in the best way. So this session is only a start. We would like to keep in touch and email us at info.fishsizeproject.org. We will send around a live document where we can share the resources. So basically, I will continue with the next speaker. And the next speaker is actually, we have a slight change in the program, which is which is Maria now, the next speaker. Thank you. 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 And maybe just a short note that we will try to take questions, at least 1 or 2 questions after each talk. We won't leave them all at the end. I won't take questions for myself now, but we will hope to have a short discussion at the end as well. Have enough time for that. So if you have questions, hopefully we can find.
Okay, thank you. Hello, I'm Marie Neal, and I'm a founding director of a small tech company called Ustum Tech Limited that's based in West Wales. We established it in 2011, and we do all sorts of bespoke equipment for environmental sensing and things like that for researchers and research institutes. So we have products ranging from precision crop spraying gear, water quality monitoring, and robotic boats that are monitoring the calving of the glacier in Greenland. But I'm here today to talk about CatchCam, and I'd like to thank Katarina for inviting me, and Natalie Holt and her team who are here from Bangor for involving us in taking this exciting research. Okay, so I'm here to talk about CatchCam. CatchCam came about around 2 years ago when Natalie wanted to automate measurements of crabs and lobsters and ultimately automate them from live onboard monitoring and processing data on board with results of those measurements straight out from onboard. So CatchCam has several modes of operation, and so it's a robust, it's easy to mount on a boat, it's waterproof, it's 12-volt power supply to it, so it's really versatile and easy to have on board, and it has several modes of operation. It's an access point, so you can control those modes through a website, and those modes are it gets triggered by motion under the camera, or it's a known distance from port and a known rate of knots, or it films all the time unless it's dark, so you can stop it by putting a cap on it. So that's the data collection. We get video, then from the video we extract images that have crabs or lobsters in them. Those crabs and that's done by a simple subtraction from a base image from the video, so that we know we've got crabs and lobsters. And then there's the next step, which is to annotate those images. So we're I'm being quick because I only have 5 minutes, but we capture the data, we get the images, and then we do processing of those images. We use VGG Via, which is an annotation tool, and in that we mark up crab carapace edges. So we get an XY point of each edge of the crab carapace, and we do lobster length. So that's one set of data. We get JSON files that we then feed into our neural network to train up the networks to ultimately get a model that goes back on board and does it all automatically. However, we have lots, as you can see, lots of different types of images, different onboard situations, different lighting, different setups. So we need lots of training data for that. And we're in the process of getting all that training data. We're also working with CFAS in England, who have got some cameras, and the Marine Institute in Ireland, who've got cameras. And we're working on getting lots and lots of data. I've also done some other AI approaches for various other, just looking at individual images, such as pixel count, OTSU, various other methods for looking at those. And we've got some good results from sexing. So we have 2 classes that we want to, male or female, and we've managed to train up networks where we've had about 50 of each class, and we've managed to do that. So it's all ongoing. We're gathering lots of data, we're processing all that data, and ultimately that will go back on board and be onboard processing. So if anyone's interested in more information, do contact me. So there I am, Marie at ustumtech.co.uk. Thank you.
Thank you. For one or 2 questions while they're changing presentation.
Real quick, if you want to screen on that.
Yeah.
Yeah.
I have 2 questions. The first is the model
Also we can hear.
the model for the determination you had
Yes.
for lobsters and crabs.
So I've done just crabs because they're easy. and because that's what I have more data for to start with. And that wasn't from on board, that was, it was from on board, but it was processed after. So it shows that it will work, but ultimately we want it to work as the fishers are processing it. And we need a little bit more data again to do that.
Yeah, just because we're talking about data and
Yeah,
I've heard a lot of, the projects that, the main challenge is to annotate data.
yes,
Find that it's a few,
yes, yes, absolutely. And we're going to spend the next few months annotating data, gathering the information, which is fairly easy now, but then annotating is going to be the slow and laborious process, but it needs to be done to train the networks.
Any questions online? No, not yet. Okay, we have time for one more quick question.
How would you manage to, where do you locate
The catch cam.
yeah.
Yeah.
In both, how would you work with different poles that they have there?
So it's a solid plastic lump, and what we've done with other people, Bang team here will know, is clamps and bits of scaffolding, and it depends on the fisher and the boat setup. They will do it. Charlie and Charlotte would know better, but I think that's how it's done. Okay.
Thank you. We realized that we need to repeat the question, so we'll do it next time. But anyway, the question was where to locate the camera. Okay, thank you.
Thank you.
And we'll be in touch.
Brilliant. Thank you.
Now the next presentation. By Lorenzo. No, I think we just walked for 1 minute, number 6. So we just go now to number 3 because Mary had to leave. So Lorenzo from WorldFish about nutrition sensitive monitoring.
Yeah, thank you. I'm Lorenzo, and I'm currently working as a data scientist for WorldFish. And the presentation of today is basically the result of a work involving 3 people. It is me, of course, Alexander Tilly, that is the senior scientist, and the main head of the projects that I'm going to present you, and Gianna Bonis Profumo, is a postdoc expert in food systems. So I'll start introducing the okay. Yeah, okay. We start introducing the talk with the main general background problem that we are trying to disentangle in a data-driven way, that is malnutrition. And it is a global issue that is also going to increase in recent years. And one of the Asian regions that is most affected by malnutrition is Timor-Leste. It is an island located in the Indonesian archipelago. And in that place, especially children malnutrition is very common. As you can see from the plot, between 40 and 50% of children suffer from stunting and anemia. That because the general dish here is just a piece of rice with some vegetables with a very low supply of animal proteins. In that region, we already deployed a digital monitoring system to monitor small-scale fishery in Timor-Leste. That is called PESCAS. PESCAS is basically a digital infrastructure where we develop to collect small-scale fishery data. We retrieve data from basically 2 main sources. One is vessel tracking movements, because we have installed GPS trackers on a defined population of fishing boats. So we have a view of the general movements of fishing boats around the island. And the other one is rely on a network of enumerators that are distributed along the island that interview the fishermen on the landing site. And they interview them on the average catch size, on the catch composition, the gear type they used, the habitat that they exploited, and many other parameters. All this information is uploaded on the cloud, and we use GitHub in order to trigger a pipeline, a data pipeline, where the data are downloaded, processed, cleaned, validated, and then exported to the public. We use three platforms to export this data on the public. We use the Dataverse, that is a data sharing portal. We use daily reports to get updated on the fishery of Timor-Leste, and we also have a public dashboard where we synthesize the main insight of Timor-Leste fishery. This is how the dashboard look like. We have like a sort of dynamic map where you can get the catch and the effort around the island, the time series of the catch, of the revenue of the fisher, and many other information. I will invite you to have a look at the pipeline, to the dashboard. Unlikely the link is covered, but it's spacecast.org. And now back to the main problem, background problem. The key question is, how can we use this data in order to reduce malnutrition in Timor-Leste? And the first step to do that is to try to use our information, that is catch and catch composition, into nutrients proportion. That was possible using the very interesting tool proposed by Fishbrain, that allows to associate to specific fish group or species the nutrients contribution. And that was based on the findings proposed by Christina Hicks and colleagues. So we integrated that tool into our pipeline. So we are now able to estimate the nutritional contribution of each catch in Timor-Leste and relate this data to the recommended daily intake. In this case, is the recommended daily intake of pregnant woman. So we can see that on average, selenium is very high concentrated in Timor catches, and is enough to satisfy here about 60 people for a single catch. It is about 7 kilograms. And on the same way, iron and vitamin A barely satisfy the recommended daily intakes of 1 or 2 people. Similarly, we can estimate what is the contribution of the most important fish groups caught in Timor-Leste. But more importantly, we can also estimate what are the habitats that are most valuable in terms of nutritional contribution in the island. So, for example, we estimated that traditional feds are by far the most important habitats to move the fishery on. So this is quite interesting, because in this way we do a first step in order to move the fishery towards a nutritional sensitive strategy. And so basically, with the collaboration between scientists, administrators, and fishermen, we can do a first step in order to reduce malnutrition for specific areas. Thank you. And please have a look at spacecast.org.
Thank you so much, Laura. One question? Or... Yeah. Maybe we'll just repeat the question, because they say you can hear it very well. Yeah.
This is for whoever wants to take it of the three presenters. My question is, this is for sure a biological problem that can be characterized from that. But I think it's also a social economic one, because whenever we're collecting this data, or whoever collects it is interacting with the fishing community, the fishers. So what goes back to the fishers, and to what extent is their knowledge in these other dimensions?
Okay, yeah, of course, there is an important step that is communicated with fishermen, because the point is, if they are aware of the problem, they of course can, I mean, they are stimulated to go fishing in certain ways. So they can, if they know that there are some malnutrition problems that they are affected, of course, in this way, they can, they are just simulated to accept some administrative strategies and so on. So, and this is also part of a work that is upcoming with PESCAS.
Is there anyone who actually gives financial retribution to the fishing communities they're working with for collecting this data?
No,
The question
they
is whether there is, I don't know.
Yeah.
The question is whether the fishing community who collects the data gets any financial.
no, they are not fishermen that are collecting data, but there are enumerators that are hired by WorldFish that interview the fishermen on their catches. So there is people hired on the field that interview the fishermen, and of course the fishermen are aware of anything. They are just accepted the policy.
All right, thank you. But I agree that this is, while we're switching to the next talk, I agree that this is a big question of who owns the data and how does it actually go back to the people who contributed in one way or another. But I think we can probably have the whole session about it, which is an important session to have maybe in the next fisheries conference. So the next speaker is Catarina Silva, and she will talk about how we could use machine learning models to get data on fish size in cases where it's a missed opportunity. And for people who are participating online, please write your questions in the chat, and we have someone monitoring the chat.
There is another question.
Not now, but we'll keep it for the discussion hopefully at the end.
Thank you. Hi, everyone. My name is Catarina Silva, and I'm a researcher at Nature Research Center. And I'm going to talk about this method we have been experimenting on to extract information from citizen science data, from images, to predict fish size classes. So as you've heard from Asta's talk, the body size of fish is very important for the ecosystem because it's closely related to the trophic niche they are, meaning that small fish eat and reproduce differently from big fish. And studies such as this one in the image have shown that fishing management practices that preserve the body size diversity in a population lead to better recruitment. Also, as you see in the bottom of the image, when preserving the fish size diversity, the population becomes more resilient to global warming, so it directly benefits fishers. But it is very, in most fisheries, there is no information about size diversity in a population. So it's often missing, which is even more common in small scale and recreational fisheries. So there is a strong motivation to conserve fish stocks, of course, and there is data available. Large quantities of data is being collected every day. In the specific case of recreational fisheries, it's very common for fishers to take pictures of their catch and upload them to fishing apps such as this one, Angler's Atlas or Fishbrain. And also it's very common to upload to social media, as you can see in the other image on the right. And if we combine scientific traditional methods of data collection with this new and huge amount of data that is available, citizen science and social media, we can validate these methods and of data collection, and we can actually use this super important information that is available on fish stocks worldwide, everywhere in the world. And artificial intelligence here can play an important role because of this huge amount of data. It can automate and accelerate data collection and analysis. But the first technical challenge we encounter with this type of data is that we don't have a reference object in the images. So I will show you an example of what I mean. In a commercial fisheries setting, what it's done usually, it's to set up a camera on the boat And the distance between the camera and where the fish go through is known. And we also have a reference object, such as the blue line you can see there in the conveyor belt. So with this information, it's possible to extract very precise information on the size of the fish. But in our case, this type of data, we don't have a common reference across all images, but we do have commonly a person holding the fish. And so a hand, we see a hand and the person. And what we hypothesize here is that maybe we can develop some models that can use morphological information on the humans and such as hands and, you know, morphology. And the model is able, of course, not to determine the precise size of the fish, but at least to give us the class of the size class. And that will be super relevant information, as you've seen in Asta's presentation. So as an experiment, we extracted data from the internet, so from Facebook and Instagram, on fish images that had metadata associated, so on the size. And we assume that this data is correct just for the purpose of this experiment. But of course, it's not good for optimal model development. And we use these 4 size classes. They were more frequent, so between 50 and 70 images per class. And the classes were between 40 and 60 centimeters, so quite wide, but we don't have a lot of data at the moment for these. But just for the purpose of these experiments, we also did data augmentation, which is a technique to increase the variability and data in general. So it consists of transforming the images, such here, for example, we did horizontal and vertical flips. So ended up with 600 images because this method has been shown to improve in general model performance and increase data. So I will show you some very preliminary results of the best model we've trained. And I'll just say, so the overall accuracy of this model is 65%, meaning that it's not good. But if we look at the diagonal values in the diagonal, this confusion matrix, you see that for class 45, it predicted in 90% of the cases, for 90% of the images, it predicted the correct class. And this was the class I had most data on. So this is just the groundwork for what we are planning to do next. But it just shows us that it might be possible to use this approach, but we also want to try other approach, of course, like object detection and other ideas. But the first steps are really to collect more data with size information that we are on viable size information to train a good model for these. And that will end up just with a call to action that we have developed this app for our project, Fish Size Project app. At the moment, it's only available in Lithuania, unfortunately, but we are planning to expand it to other countries. And we would love to hear from you if you have data, this type of data available and would like to collaborate with us. So we have our contact details here. Please get in touch, and thank you for listening. Yeah.
It's a very interesting approach, but I think it's not so much scientific because I need to take the measurement of the fish, including photos on our flow with the etc. The other approach with references, it's very easy to convert the size according to the reference. But most of the fishes I saw in the found in the we don't have some references. Also, take into account that the fishes that are taking the telephone together, then filters that it's not the real size of the fish. I mean that the size of the fish is in the front and it's bigger than the person, so you overestimate the size of the fish. But if you get the size of the fish, you can convert it into weight, it makes the weight of the medicine. So you can get more accurate data. If you get length, you can convert it into weight, you know, the species. But take a scientific measurements from photo, from film, I think it's very risky to get an accurate research information.
Yeah, maybe, I don't know if.
So that's over
you
there.
hear?
Now I get it.
So I have to repeat. Well, basically, the question is, or the statement that this is not scientific and that we should not trust the data that we find on social media.
Mhm.
Yeah,
So, yeah,
Because the people hold fish close to the camera, far away to the camera, and we don't know what the size is.
yeah, so first of all, to address the fact that the size, it's not correctly measured, for example, for a proper model, we will use data that is validated. So with known correct measurements. This was just for, as I said, a groundwork, a pilot study, just to validate if the method might be possible. And in regards to the fish being sometimes close to the camera or far, so maybe I'll start with some, methods that are scientifically widely used everywhere, such as underwater surveys. For example, divers count and estimate size of the fish underwater. I have been doing this for a very long time without a reference object, and they are really good at doing this. They get trained, and they estimate very accurately. There are a lot of scientific data on that. And in this case, I know that if we are using a human face, for example, it might be in the images might be very different. But if the model is somehow using also the hands are also the same level, at the same distance from the camera. And if we use a good variation of the position of the fish, or you have to give all this information, all this variation of data. So you need a lot of data to train a good algorithm. And of course, we won't have the precise measure of the fish, but we need to know how far can we go in the granularity of the size class. That is
one more question online. Yeah.
Saying very interesting job. You use deep learning, correct? Or which other method? And how do you measure the human to make the scale?
Is it deep learning or some other methods? And how did we measure the human to make the scale?
Yeah, at the moment, so I'm not trying the object detection. It's deep learning. I'm not trying the object detection, so I'm not measuring any object in the image yet, but I will try that approach as well and compare with this one. So this is an image classification approach only. It's just taking the whole, all the information in the image, so taking a full matrix and letting the model filter and extract the important features for determining the classes. So it's a classification problem.
I think we'll have to go to the next one, and that's a very quick one. Yes, one more.
No, this one will be possibly the expert that data from the camera in EXIF, that is development on focal length.
Whether it's possible to use like a focal length and the distance to the camera. Yes, I think that would be possible.
Yeah.
If we had that information. If you go to social media, you don't have that information associated with the image, but it would be good, and that would be useful. And so with this, we will go to Nicholas from from Fishbrain, about also how Fishbrain is doing fish species identification.
Yes, cool. Thank you. Yeah. Yeah, I guess I will sit down here. Cool. So I feel like a teacher now. It's cool. Cool. Nice. Yeah. Hi, everyone. My name is Joar, and I'm a data scientist at Fishbrain. And I'm going to talk to you about how we can use kind of deep learning to identify fish species from photos. And kind of more specifically, I want to talk about how we can make like these deep neural networks more explainable, and so we can know a bit what's going on inside of them. But first, sorry. How do I go? How is it touch screen?
And now you can just hit.
Right. Okay, good. Cool. So yeah, Fishbrain. Fishbrain is an app for sports fishing. We're both like a social network where you can upload your catches and kind of interact with your fishing friends. But we also supply like utility functions where we kind of help the angler know like where to go fishing at what time and with what gear. And we have 14 million users and we have 12 million catches logged. So we have a lot of data, and I want to talk about like one of our specific features that they kind of leverage these huge amounts of data. So we have when you upload a picture of a fish, we have kind of automatic like species identification that is powered by deep learning, which is, as most of you probably know, like a subset of kind of machine learning methods, and it's based on these artificial neural networks that take inspiration from how the human brain works and kind of the synapses there. So the idea is that you can train such a network to take a photo of fish as input, and then we'll get like a label as output saying which fish species is in the photo. And one of the kind of points of critique for these methods is that they can be a black box, meaning you don't really know what's going on inside of them. And so that's what I kind of want to address today. I want to show you like one method that I use to kind of debug my neural networks a bit and understand what's going on inside of them. So I've looked a lot at this tool called FastCam that's developed by some people at Cornell University. And this method, given a neural network and an image, this method finds like which parts of the image were most important for the neural network to make its decision in that specific case. And so I'll walk you through an example of how this image of a pike and this ResNet neural network, when we apply FastCam to this network and this photo, and we'll see like which parts of the photo are most important. So this ResNet 50, it has like kind of 5 different stages where we kind of upsample the photo to 5 different sizes. So using FastCam, we at each such scale, we compute like a heat map where you can see like which parts are, so the white parts are deemed more important for the model and in making its decision, and the black parts are less important. And then we can sum all these together, and we'll get kind of like a summary of what the model looks at across all of these layers. And then if we apply like this color scheme to this heat map, where we go from blue, meaning like blue violet means that the early layers of the network looked a lot at this area of the photo, but on the other side of the spectrum we have like green and yellow, meaning that the later layers looked more at this part of the picture. So there we can see, and if it's white, it means that all kind of layers looked at this area of the photo. So here we can see like how these each kind of individual layer heat maps were combined into this more general one. And then we can also, if we take this combined one and we visualize it in a more kind of intensity heat map, we can see how these parts that are deemed more important, how they really pop. And then what FastCam does is that it takes another heat map which represents kind of which parts of the image were most strongly associated with the output class, and then you multiply these heat maps together, giving you what you see, the heat map that you see at the bottom here. So that's kind of like the final result of the like FastCam tool. So then in this case we see that, okay, for this image we look, the model looks a lot at like the jaw of the pike and also the fins. So this can be kind of a good tool to use if you like, maybe you're in a stage where you've trained your model and you like evaluate it on your test set and everything looks good, but you still want to see like, okay, what's going on inside there. And then you can use this tool to kind of get a peek into what's actually going on inside the network. So I really like encourage anyone working with this type of stuff to have a look at the FastCam paper and the code. It's for me, it's been really helpful. And finally, I want to share like my contact information. You can please contact me on my email if you want to talk about this kind of stuff or just fishing in general. And also, I'm on Fishbrain. I'm perch face, so follow me and you can see what fish I'm catching. Yeah, that's yeah. Thank you for listening.
I didn't know you can look inside the black box.
Yeah.
To be honest, you can actually, now there are some, okay, there's some questions. There are questions?
Yeah, Thomas White, first question is, how many different fish species can Fishbrain AI model detect accurately?
How many different fish species can you
Yeah.
detect accurately? What is accurately? That's a, that would be hard, so yeah.
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 So we have with the model we have running in the app right now, we have 80 fish species.
80?
Yeah,
Yeah.
80.
yeah.
Yeah. So and yeah, accurately is yeah. Yeah, but I guess it's quite accurate in general, but there are some species that are more difficult, like salmons, for example, or like I guess it's quite easy for a model to know that it's some type of salmon, but then it's really hard to distinguish between like yeah, yeah. But our ambition is to have way more species though. So that's kind of what I'm working on now, to release like a new model that knows like several hundreds of species. So stay tuned for that.
The next question is, how many images per fish species are needed to image the
How many
Yeah.
images per fish species you, I guess, in your case?
Yeah, great question. It's so what I do is I kind of pre-train a model so that it's trained on like this big standardized data set of like things that are other things than fish, like buildings and dogs and whatever. And then I kind of fine tune it on fish data. So what I found is that it can be sufficient with like very low, quite low amounts of data. So like, for example, in the order of magnitude of like 100 or maybe even 50 per species can be enough.
So there is one question here. Maria had a question.
If you're looking inside the black box, are you then using that to decide on what layers you're doing? Are you then fixing the network?
Oh yeah.
So if we're looking inside the black box, do you actually then modifying the black box, or is it just to know what's happening?
Yeah, no, right now it's more just to know what's happening. Yeah, good question.
Still the black box in there.
Yeah.
I think we probably need to move to the next talk, and we'll keep those online questions for the end. Hopefully we'll have. And so the next speaker is Mocha. We wanted to say, and so Ahmad Katur with a Mocha from University of Tasmania, also talking about electronic monitoring in a small-scale tuna fishery.
Hello everyone, I'm Moko. I'm PhD student working on the electronic monitoring as well as for artificial intelligence to analyze the data from this electronic monitoring in the small scale fisheries. So let's get started. How to do that? Okay. Okay. So yeah, in the background, small scale data is at the moment is in the state of data poor or completely absence. We know all of that, and it's quite challenging to obtain of the catch data. And it's going to be like a more challenging as well to apply electronic monitoring in this type of fishery. And to give you idea on how small the fisheries that I'm working is only 1 gross ton, 1 day fishing trip, and a single crew, no power or something. So it's going to be like challenging environment. But these fish are actually are fair trade and MSC certified fishery, so it's quite something. And the objective of this one is actually not applying electronic monitoring for the enforcement or something like that. Is to obtain all the catch data. So identify the number of individual, length, weight, species of fish recorded by the camera, and also its coordinate location of the fishing ground, and also compare it to the conventional pod sampling data collection. So to achieve this one, I'm using low-cost camera. It's only $100. It's actually an outdoor CCTV camera and also low-cost GPS tracker. So at first, I put the camera in the front of the boat, and then from the second camera dedicated for the species identification, I just put in the side before the fisher put it in the cold storage box. So they usually use this kind of deck for the wash the fish before put it in the box. And here is a bit about the artificial intelligence thing that I apply. So I'm using YOLO version 4. And then it's trained to identify 2 different species, main target species of this type of fishery, yellowfin and skipjack tuna. And it's run on the Google Colab Pro. It is only $10 a month. So the key method that I want to emphasize in here is I need to build something that is easy to use, both of the fisher and also the fleet enumerator, and also not disturbing the fisherman work because it is a limited space on the boat. And another thing is it has to be cheap because we have to build something that is low cost or even no cost, because you cannot build something like $1,000 system and apply it in the small scale fisheries. For the economic point of view, that just simply doesn't work. And we also collaborate with the local NGO. And by the end of the project, we have to move everything, the code and everything, transfer of technology to this type of NGO. So at the end, they have to make sure that they can run everything, the system, the AI by themselves. So most of them are not programmers as well. And here we go. So on average, we have like a 10 hours video duration for this one type of fishery, and the AI is work scanning frame by frame, and the camera is working on the 15 frame per second, and we have like this so how do I play the video? Okay, so just move the video.
You're trying to do.
Yeah, just play the video. Okay.
You might have a little trouble with the.
Okay.
Just let's go to here.
Okay. So yeah, you can see in here the YFT is a yellowfin tuna, is having the class. The 6 is the individual number, and the last is the confidence score. Is going to be going up and down depending on the angle of the fish relative camera. But we tell to the machine whenever the confidence score at the highest maximum, so extract this image from the video, and so at the end we have like a nice table with everything this from the video. So, and it is a raw video, so just raw video. We didn't do any calibration or modification to make it everything simple. So just fit in the model, and at the end we have a nice table of the list of the fish that is recorded on the video. And here is the result, example result of the still image that is extracted from the model. You may notice that there is 3 small square black spots in here that we use later for the reference point for the length measurement photogrammetry. And the model is also working for the smaller one, so it can distinguish between the yellowfin tuna and also skipjack tuna as well. So it's quite good actually. And remember that we have a GPS tracker as well that we attach on the fisheries. And if you might notice that there is a time attached on the every image. So by simply extracting the time, we can and match it with the the GPS data, we can simply get the exact fishing ground of this every single fish that is monitored in this camera. And the GPS is working pings every 5 minutes, so it's quite really close accurate for this type of series. And this is for the length measurement. I use like a reference point for this one and measure it the fork length of this. But this is something that I need to improve in the future. So at the moment, we don't do any calibration or something like that. In short, it works, but it's not the way that we want to, because next time we are going to use stereo camera or LiDAR. So instead of you getting the length, we will get the exact 3D-dimensional shape of the fish. So that's going to be like a better for us to get everything, the data that we collected. And lastly, of course, if we get the length of the fish, just multiplied by length by the relationship, we can estimate the weight of the individual fish. And getting everything this from the video, directly put it in the model, and at the end of the day, you have nice table. Is going to be like we so many opportunities that is we can use on this technology. And I think that's for it. Thank you.
Thank you very much. Looks promising and exciting. So we have one question here.
I have, but I'll try to be short.
Just one.
First was, how many additional costs do you need to analyze these 10 hours?
Yep. Okay.
So how many hours of analysis computation do you need to analyze 10-hour video? Mhm.
In my case, the camera is working at 15 frames per second, and the model is able to do it up to 32 frames per second. So it's gonna be like a half that we need of the video. So imagine that we have 10 hours, we only need 5 hours to analyze that. But we also use a parallel computer as well. So instead of 10 hours, we can chop it 2 hours, 2 hours, 2 hours, and run everything at once. So at the end, we have like a 30 minutes or something like that.
Okay. Any questions online? Right. I think we probably need to move on, but I'm hoping this will spur a lot of further discussions that we can continue during lunch. And so the next is joining us online, but to facilitate a more smooth presentation, we will be they recorded the presentations. So we watch the video, so we don't have to worry about screen sharing and so on, but they're attending online to take questions. So the next speaker is Andrew Halford from FAME, Pacific Community, about development and integration of e-data system for coastal fisheries management.
A quick hi to everyone. My name is Andrew Halford, and I'm a senior coastal research scientist at the Pacific Community, commonly known as SBT, based in India, but working throughout the Pacific Islands and territories. The presentation today is about a renewed approach to data collection for supporting coastal fisheries management, which we've been developing and introducing to our member countries over the past few years. This slide represents the world that we inhabit, and this is really all about working with Pacific Island countries and territories to enable them to sustainably manage their coastal fisheries. The reality is this cycle is chronically stuck within the first 3 checkpoints. And recent multiple global scale analyses of coastal fisheries health have also concluded that there's not enough information to assess the vast majority of stocks in the Pacific region. So this is the reality. Now, while there are multiple reasons for the existing issues in coastal fisheries, it should be recognised that coastal fisheries is an incredibly diverse system in the Pacific, and there really is no one-size-fits-all approach. Slide just demonstrates the sheer range, or the vast range of resources that are harvested in this world. Capacity and research for institutions struggle to capture sufficient information across such a range of these resources. So the question is, how do we break up this status quo and clear these roadblocks? I want to use a juggling man metaphor here to explain the approach we've taken, and really the way we see it is the most desirable fisheries management system is often the most complicated in terms of the data required, expertise of the personnel and the resources needed. And in the Pacific Island countries and territories, this leads to information overload and swamping of capacity, essentially too many balls to juggle. So by recognising what the inherent capacity is within small island developing states, we've developed a system with relatively minor data collection requirements. Link this with a web and app-based electronic system, which provides a simple but powerful system for data collection and reporting in coastal fisheries. The time saved is significant, and this has already been used to increase sampling across the country. We've created what we call a data collection spine, which is really just the foundational layer in what is inherently a hierarchy of the system. But the idea is that further complexity can be added as and when possible. This really brings me to the fancy bit, if you will, of this presentation, and that's what we call the gateway to this system. It's a web-based coastal fisheries dashboard, if you like to call it, with direct links to the data collection modules and aided by an app called Ecosavia. And this is essentially the electronic data sheet for collecting data. And we have a range of modules there and they can typically develop as needed. What we've done is we've adopted a photo-based system for collection of data where possible, and we've linked this to a powerful artificial intelligence system to automatically measure and record the length, weight and species of each individual photograph. This capacity is extended from birds as well, although I'll have to say that the sheer range of size and three-dimensionality of invertebrates has required considerable more effort. As you can see, we have to use coin, which gives us something with no dimensions to enable us to calibrate measurement errors caused by the height of the animal. Once you start collecting this information and you've got good information electronically captured, this system becomes a simple but powerful tool for quickly interrogating data patterns and reporting. Here's just a very small example of plotting the size frequency of a particular species, and over the top of that we can put biological reference points. Quickly ascertain whether overfishing is occurring, for example. Length weight plots also identify dubious data points to ensure analyses are not contaminated by such data. And I'll just make some calls about, for example, this fish, 50% of the fishing markets are undersized, and so it's the measurements are as too dubious. Along with the graphical analyses, we obviously have ability to table data, and I think this provides equally powerful insights in the fishery. We've given two examples here. For example, 50 calculating the percentage of fish under minimum legal size and species. So looking at a summary of sentiment across markets, and you can see just those two tables in themselves offer a huge So where does it leave us? Well, we're a small team, so the pace of scaling up is constrained by the reality. However, we received overwhelming endorsement from members.
Very much, Andrew. And I think Andrew is online, so yeah, here we go. We can just see.
Yeah. Did you guys have any better luck with the volume where you were?
We could hear you very loud and clear. Was it hard for you?
Okay. No, I couldn't. Online was terrible for me, but I don't know whether that's reflective of everyone or not.
I'm not sure. So how did the others hear online? I'm not sure because anyway, all right. So do we have some questions in the audience? I do actually have a question, and the question is, so how many like fishers, and so what's the size of the, like how many people you are actually interacting and working with in this way in terms of data collection?
Well, for the most part, it's been mostly market-based at the moment, because that's where the bulk of the fish in the countries that we've been working closely with, mostly, it's mostly markets, but we're set up to do exactly the same with individual fisher work as well. So it's very much dependent on the countries we go to. You know, a lot of the larger countries have centralized markets, and so to get the most bang for your buck, you want to go to those locations because that's where the vast majority of fish that are being caught are coming through the system. So that's where you want to capture the information. But certainly some of the other countries which we're moving into now that COVID has sort of slowed down enough to let us to travel again, and some of the smaller countries that don't really have big centralized markets. So in that case, we will have a lot more information being captured by the local fisheries officers that are more an individual fisher interview one. So at this stage, it's mostly market-based information, but there's no reason why the fisher-based side of things won't become larger as time goes by.
All right, thank you. No questions here. Okay, not for now. Thank you. The next talk is by Jeremy Prince and from Biospherics about how do we analyze the data that we have. How do we use the existing data? So we try to launch this.
Thank you for this opportunity to talk about how we are using community-based science to inform and energize small-scale fisheries reform in the Pacific. With initial community workshops, we enable people to see for the first time that the changes they are observing are symptoms of overfishing. So they begin asking, what can they do? The solution to overfishing requires changing old ways of thinking. Before our workshops, people mainly think about not wasting fish. The smallest fish are sweetest. No one releases small fish to grow and breed. That would waste good food already caught. Heuristic thinking that is now unsustainable. We challenge that thinking explicitly by contrasting with nurturing gardens. The reef is a garden and the sea. Eventually, you can't keep harvesting if nothing is being sown. Which brings us around to how much breeding is enough. First, we explain the human reproductive index. If on average, each adult couple has about 2 surviving children, population is stabilised. This is the replacement level. Below the replacement level, population decline. Spawning potential ratio is equivalent. Without fishing, fish complete 100% of their natural spawning potential. Fishing shortens lifespans, reducing spawning potential to some ratio of the unfished level. From studies, we know the replacement level for fish is about 20%. Willing communities are taught to collect data and see for themselves the extent of their own overfishing. The main data needed is catch size composition. Estimates of size and maturity are also needed initially. It turns out that teaching fishers to macroscopically classify gonads empowers community management. Whole communities start seeing and talking about how all the fish are being eaten before breeding. Once seen, it can no longer go unseen. We initiate and support community sampling programs as beachheads in each country, embedding local fisheries agency staff with them. In this way, we develop local change champions and communities of practice. Across all levels of society, we give people a common language about overfishing, facilitating society-wide discussion about stopping. Our results galvanise dialogue. Everyone knows it's a problem, but putting that SDR number on it increases the urgency. Here are results from Solomons. The blue showing size composition at 100% without fishing. The white 30% SDR. The red is our sample with which we estimate 27% SDR. The results translate simply into management prescriptions. For example, multiplying size and maturity by 1.2 approximates a good minimum size, which can prevent recruitment and growth overfishing. Which can prevent recruitment and growth overfishing. Our message to communities is that overfishing is catching the fish before they replace themselves. Effective management allows fish to grow into adults and breed sufficiently to replenish the reefs. By taking note of fish sizes, communities can see for themselves what is working or not. Everywhere spawning potential surveys have galvanised management trials. As this story shows, even without the support of central government, community management can still be facilitated. I've only met Matthew Merrick in PNG 3 times. Here he is with his handmade canoe and fish measuring book. He came to our workshop expecting to learn how to catch more fish and left angry at the end of day 1. But on day 2, we changed his world by teaching him to look inside fish. Home again, he trained and organised a team of 6. Over 2 years, they measured 30,000 fish. With his data, he defined the spawning season of rabbitfish, his village's most important species. Then he persuaded his elders to implement a daily bag limit for the season. When the season was poorer than expected, they implemented a 3-year moratorium instead. Today, Matthew is a barefoot ecologist with a mission. Before, he only wanted to catch more fish. Thank you.
Thank you very much, Jeremy. It's a very inspiring presentation. And yes, and we can see you online now here as well for us. So if people have questions here from the audience or from online. Yes, there is one question.
That's from me, actually. No, I can't hear.
A question from Niels here in the audience. Yeah.
Yeah, that was a great talk, first of all. And so I'm wondering how certain are we about this 20% threshold for population stability and whether there's any good empirical ground in it.
Yeah, I'll have to repeat it, I guess. How do we know it's 20%? Like what sort of data we have on that and what sort of ground? How certain are we about? Could it be and how different could it be?
Okay, so the theory behind SPR was developed back in the 80s, 70s and 80s in the Northern Hemisphere in the North Atlantic. And there's been a lot of work done on both assessed fisheries as well as theory to come to this 20% number for teleos, at least experience is it's actually quite a conservative limit reference point. So the point below which you run into a risk of recruitment declining. So that's quite well established in science and accepted international law and by certifying bodies, Marine Stewardship Council. But it is known to be a generic reference point. And with MSC, if you have the evidence from your own stock assessments that it should be varied to that, and normally it's varied downwards, then you are permitted to go to your empirically proved or analytically proved level. It's also very much around teleosts. It's accepted that lower productivity taxa like sharks and things will need higher reference points. But I think the major point, rather than getting hung up on what is the right number, is that it actually, for the first time, for the extremely data poor fishery, gives us a metric of management that can be compared to something rather than just trends in catches or income per day or whatever, just our average size of fish. Those can tell you directions of fisheries or indicate directions of fisheries, but they don't give you some hard reference point to communicate with and recommend management with. And sorry, the 1.2, the size limit 1.2, that's based on modeling Adrian Hordick and I did and published in Fish and Fisheries. So that's the idea that generically 120% of L50 is the size at which you are protecting something like 20% of SPR. And by the way, you optimize your catches at that level as well.
Thank you, Jeremy. I think that's a very good answer. And I would like to discuss that a lot more. It's a pity you couldn't come to this session, but I'm hoping we will continue this discussion later. Sure. At the moment, we need to move to the next talk. Thanks. And this is a slightly different one, but it's very interesting how we could use machine learning to estimate fish abundance from individual recognition.
Hello, everyone. It's good to be with you. My name is Fan Hipp. I'm a fish biologist with the U.S. Geological Survey here in West Virginia in the Eastern Ecological Science Zone. And I wanted to talk to you about some of our recent work in AI for the purpose of abundance estimation via crowdsourcing and citizen science. So I appreciate your time. Let me just get right into it. I want to first thank co-authors. This is a collaborative effort. Zhonglang Zhao at University of Georgia, Weili Shi and Sheng Li at the University of Virginia have been instrumental in developing some of these analytical pipelines. Also, Ben Letcher with the Eastern Ecological Science Center is a collaborator in this effort. So here's our vision. We imagine a system for enabling anglers to understand the history of their catch using individual recognition in freshwater ecosystems, and this could be a game changer. Obviously, there's been a legacy of this work in marine systems. There's been a legacy of this work in terrestrial systems, but really in the freshwater, commercial and recreational fishery space, this is new. And we see an opportunity here. We have a proof of concept that I'll be talking about today, and then a look ahead into where we think this can go. So the steps are essentially as you see here, anglers providing images of fish they catch, submitted through an online portal, an AI platform to do individual recognition, and we treat this as a virtual mark recapture experiment study, really, to identify abundance and to estimate movement and to estimate survival. Okay, so that's the vision. So we're going to be talking about brook trout. Brook trout are a native species in eastern North America and are extremely culturally important, economically important, ecologically important, and for the purposes of this word, they are highly spotted. So let's take a close look and ask the question, do individual spot patterns enable individual recognition through these AI pipelines? And if so, we can treat that as a market capture study for the purpose of abundance. So when we zoom in, we look at the side of brook trout. This is Savalinus, is the genus, the char. We see that there are all sorts of spot patterns. Anecdotally, we kind of recognize these things as potentially being individually diagnostic, but that's never really been tested. So that's what we wanted to do first. So the analysis pipeline that we'll be talking about, I'm not going to go into detail with this, but to give you a general overview of it, there is a fish detection stage, identifying the fish from non-fish in these images. Then there's a feature enhancement stage, and this is important both for a region of interest and for screening, essentially a filtering process to identify the key features, the potential thumbprint of these individuals in an AI context for the purpose of individual identification. Okay, that's essentially the analysis pipeline, and you can see it laid out here. So through the submission of images, extraction of fish from non-fish, and then highlighting individual components of interest that enable prediction of individual space. There's also an alignment step in there as well. And you can contact me for details on the background of this work if you're interested in that. So training data that went into this, hundreds of images using a GoPro camera from a hatchery in Virginia, where we are confident on individuals being distinct in this data set. So developing this training data set was important for us to validate the potential for this tool to work. And really, I want to highlight that it's quite accessible. The training data can be collected by anyone. We've done it in a controlled way as a starting point, but really anglers and all of the innovative crowdsourcing platforms that are now becoming available could enable this in a massive scale. But the starting point, we're doing this in a controlled way. It's a very simple design. You can see the GoPro camera mounted here above a fish board. We just move fish through and take photographs of each individual. So one of the main features and results from our preliminary work here is that the extraction of spots through a filtering process is extremely important and enables individual identity through the spatial configuration of the spot patterns. You can see an alignment step as well. And we can see that in this demonstration, we do see separation among individuals. So this works for the purpose of individual ID. Now, let's treat it as a virtual market capture experiment. And this is where we can bring the power of wildlife biology, capture, recapture studies, the field of abundance estimation to bear on this. And we can treat it as a virtual market recapture experiment for the purpose of abundance estimation. This is where we're going. So looking ahead, we will be developing an empirical validation study with collaborators in Japan, looking at white spotted char and macerated salmon. And then we will be developing a collaboration with Trout Unlimited and wildme.org to enable anglers to provide images for the purpose of understanding, has this fish been caught before in fresh waters? And for the purpose also of us to be able to do abundance estimation in an unprecedented way. We're also looking at video applications in the future, so stay tuned for that. So here's my contact information. I want to thank you for your time and I look forward to your ideas on this. Cheers.
Right. Well, as far as I know, and it's not online because it's a really bad timing for for America. And the next speaker, something like 3 a.m. at night. So the next speaker is also it's from Canada. And he is like a founder of a fishing app, which is widely used in Canada. Again, it's focused on recreational fisheries, but we included this talk because we thought that it's a wonderful example of how that could also be applied in small-scale fisheries. So how to collect data from a diverse set of fishers and how this data could be used.
All right, good day. My name is Sean Simmons, and I am president and founder of the Anglers Atlas and MyCats. And today I'll be doing a presentation on how a mobile app, MyCats, can help small-scale fisheries generate big results. So there's two parts of this talk. The first is I want to review the evolution of MyCats as a tournament platform for fisheries research. And second, talk about three specific examples of how this new approach of a tournament platform can benefit fisheries through bass battles on Newman Lake, Iowa Walleye Challenge, and then Biloxi Bass Bonanza. So MyCats launched in 2018 as a citizen science tool and a way to really see if we can collect standardized data from anglers that could replicate a real survey. That really was the first objective. We just wanted to see if we can collect where people fish, when they fish, how long they fish, what do they catch, so we could see if we're getting similar or different data than standard creel comparison. Now, since that time, the app has evolved considerably, and a key part of that was figuring out how to motivate anglers to start using the platform. That's always a significant challenge of motivation. And so we added various features. The example on the left of the screen shows the map view on the app with marine protected areas highlighted. They're called rockfish conservation areas. Thinking that this would be a way to incentivize anglers to use the app and then also contribute their catches. And it worked a little bit, but not enough. And then COVID arrived in 2020, disrupted a lot of what we do, and also presented a unique opportunity. And that is because we have two strong tournament anglers on our team, and we noticed across the country tournaments were being canceled due to COVID closure. And we thought, well, we've got an app that already catches and collects a lot of data on fish catches. Why don't we modify it a little bit so we can run a catch photo-rich tournament? And one of the benefits as well of this is that it's a strong conservation model. So the fish is caught, measured on a bump board, picture taken, and ideally in less than a minute, the fish can go back right to where it was caught. So certainly increasing the chances for survivability. Now, it's important to highlight what the key advantages of this strategy are from a conventional angler log diary or app, which is what MyCats was originally based on. Things that are problems with the angler log is the motivation for commitment, figuring out how to motivate anglers to report their catches. Also, you get variability in reporting, missing information, and it wasn't easy to incorporate other scientific research. You had to modify the whole platform to make that work. Whereas the tournament model, and some people don't like the word tournament, so called the BioBlitz or called the challenge, but it's essentially the same model that underpins the technology. And that is, there's a clear motivation. People sign up for little prizes, bragging rights as well. There's a consistent data collection expectation based on the rules of the event. So if your catches don't measure up, they get rejected. You don't get qualified for prizes. So we found that angler behavior can be modified significantly so that it provides consistent data. And probably most importantly is the ability to rapidly execute new scientific research by adjusting the rules and the prize structures and categories of prizes on the app. So first case study is Bass Battle on Newman Lake. It plays August 1st and 19th. It was built up for the American Fisheries Society conference in Spokane as a way to demonstrate how a tag tournament could be used to estimate the population of largemouth bass in Newman Lake. That was the scientific goal. We partnered with Department of Fish and Wildlife in Washington, tagged 218 bass, as shown by the blue histogram, and then ran a tagging tournament where we were able to collect data on, I think it was over 1,500 catches. And we've also got a Kolmogorov-Smirnov test to see how similar or different the data is. And as you can see, there's certainly some variation, but it's fairly tight along the way. Now, What did we end up estimating? Well, by the end of the event, 11,271 fish plus minus 4,000. Did notice a steady creep upwards, and we're going to look at that a little bit closer, but really an interesting way to estimate a population using a tank tournament. Now, a different application of this tournament model with the Iowa Walleye Challenge, where we're helping the state get a sense of what the walleye fishery was like across their whole state. They invest a lot of resources into the walleye fishery, but are limited resources to monitor what happens. So this is a way to supplement their ongoing field survey. And we didn't get a lot of anglers, 70, but we were able to get a decent spread across the state that certainly matched with a lot of the expectations and a lot of data on lake distribution, which I don't have time to get into now, but really interesting. And finally, this is Blotchy Bass Bonanza. It runs across the US and Canada until end of November. And the purpose here is to identify the distribution and prevalence of blotchy bass, which is a recently discovered virus that infects the outside of black bass. And so here's 3 examples on how we can apply the tournament model to achieve very specific management and research goals. Anyone has any questions, please reach out to me. My email is below, sean@anglersatlas.com, or give me a shout. I look forward to hearing from you. Thank you.
As far as I know, Sean is also not online because of the timing. So yeah, we can't take any questions, but I'll hope we will be able to contact each other later. And so the last talk in this session is from a FishL initiative that is started in the United States. It's a global initiative, and it's about the attempt to, it's an initiative to build a machine learning model, artificial intelligence model to identify all fish in the world. So it's a good start.
Welcome to the Official.ai presentation on how we are enabling facial recognition. Meet our team, Tom and Jane Y, founders of the Y Foundation and main supporters of the Official.ai project. They're also the creators of the mobile sport fishing platform, Fishbrain.
Sorry, the speakers got disconnected. We'll play it back again. Yeah.
Thank you.
So we can probably start from the beginning.
Sure.
Welcome to the Official.ai presentation on how we are enabling facial recognition. Meet our team, Tom and Jane Y, founders of the Y Foundation and main supporters of the Official.ai project. They're also the creators of the mobile sport fishing platform, Fishbrain. The biologists, Christina Acevedo Brazil, Kimberly Demashaw, Santiago Tricati, and Guy Urel. Our marketing department, Dario and Joy Flexer. Our current partnerships are with the Y Foundation and fishangler.com. This project was originally slated to be part of Fishangler. However, Tom and Jane Y found it would be more beneficial to the science community if this was offered as an open source tool. The Guy Harvey Ocean Foundation, Microsoft, Coda Head, and Conservation Expo. Our mission, our goal is to accelerate the research and development of a highly accurate fish identification technology using AI to recognize all fish species worldwide. Our vision is to act as a central hub for the combination of various fish image datasets into a single open source AI ML ready labeled fish image dataset that will contribute to the creation of facial recognition. Our goal, provide a specialized web portal for the collection and ML tagging of fish images. To publish the world's largest labeled fish image database for AI ML for commercial and non-commercial use. And to seek the development of a highly accurate AI model that can identify fish species worldwide. We are accomplishing our mission through 2 main actions. Our open source labeled fish image database that will be a tool for others to build AI technologies as well. And a citizen science portal that will be a web-based portal that allows users to upload, tag images with fish species names, and tag polygons. Our open source labeled fish image database came about because we noticed a limited number of labeled fish images available as open source when we began our journey. So we set out to create the world's largest. We knew that it would require a few things. It would require images from all over the world. It will require the development of a specialized website for photo repository. And it will require the ability to allow copyright data to set images submitted to the database. Including, but not limited to, internal use only and not for open source data, or open source friendly. Our citizen science portal is going to be the way we build our image database. It allows for individuals or teams to register an account. You can upload fish images easily through drag and drop, a computer upload, or a CSV file upload. It has a specialized ML editor that allows for polygons to be set around each fish in the image, as well as each trait of each fish in the image, and labeling polygons with fish species. The citizen science portal also allows for specialized data collection for each image, including GPS location, fish species within the image, and metadata. It has the ability to complete copyright data customization for each image, or you can set it for a group of images. The team setting allows for the team to review images before submission to the model. And our QA system allows for the biologist to verify image prior to final submission to the model. We have a few hurdles to success. Image acquisition for lesser known fish and non-game fish. Copyright and open source access to images. Scraping images from social media can be very beneficial, however, it's usually against their copyright. A way around this would be very useful. Images that can be freely used for commercial and non-commercial use. We're limited to manpower to label each image for fish identification and manpower to QA images before submission into the model. We can only do as many as the number of biologists we have. And our last hurdle to success is gaining momentum within the citizen science portal. How you can help us. We need help gaining traction within fishery, research teams, and NGOs in terms of getting them involved in sharing their image databases with us in the citizen science portal. You can assist us in labeling the unlabeled fish images and/or QAing images that have been submitted to the model. And funding to help contribute to this effort would go to coding and web development, building our QA team, and building the fish image labeling team. Thank you for watching this presentation on Official.ai and how we are enabling official recognition. If you'd like to learn more about our project, we encourage you to visit Official.ai. If you'd like to email us with questions, please feel free to reach us at support@Official.ai. Thank you.
Thank you very much for the presentation. I don't know whether Tom was online, whether there is, Christine is online. So yes, hi. So if we have any questions, we are sort of close to the end of the session. Always the last presenter gets a bit disadvantaged, but we still have time for, yeah.
For the database, are there any species or are they localized?
So, yeah, so the question is where is in your database, do you have any European species? Are they all American species? Where are they from?
No, we do have a European species in our our AI model is focused on the top 100 fish being caught in the Fish Angler app right now. We're kind of using that app as our gauge to what fish species need to have an AI model ready for them as soon as possible. So we have the top 100 that is a worldwide app. We do have European species as well as North American, but at the moment it's a little bit heavier on the North American side.
So I have one question. So as I understand the model and the images, they will be publicly available, right? So basically that can be used by others.
Yes, yeah. So it's this project is kind of two parts. So it's the image database so that others can use these images to build their AI projects or machine learning projects, and then also the AI model to identify fish species. So yeah, and we do have, so yes, I guess the answer to the question is yes.
Yeah. Thank you very much.
You're
And
welcome.
again, this is, we will send an email around if you actually want to be included in this group who are interested in machine learning and digital data in new age. Let us know, contact us. We'll send a document around with sort of resources and continuation. And yes, I wish we had more time for discussion, but it's lunchtime and people are hungry. So I think everybody, thanks for joining. Bye.